Three. Hello and welcome to OSU's Virtual Teacher Professional Development Series. My name is Jay Well. I work with pre-college programs, and today we have an exciting lesson for you called Bloom in a Bottle. We developed this lesson with Dr. Stephen Giovannoni's lab, and today we have Dr. Chris Suffridge, a postdoc in that lab, to really explain to you the connections of the activity to the research that they're currently doing. As always, I like to start these sessions out by making those connections to NGSS. We'll be focusing on planning and carrying out investigations as part of our science and engineering practices. For disciplinary core ideas, we'll focus on LS2, ecosystems, interactions, energy and dynamics, and for cross-cutting concepts, stability and change. But I'd like to comment that there are a lot of different ways that you can connect this lesson to NGSS. And I really think focusing on the science and engineering practices and really building skills with your students, there is a great connection for this lesson. Asking questions and defining problems, developing and using models, analyzing and interpreting data, engaging in argument from evidence. All of these are really easy to connect in the Bloom in a Bottle activity. So as you go through this and listen to this virtual teacher professional development, think about different ways that you can bring science and engineering practices, as well as disciplinary core ideas and cross-cutting concepts into the lesson. Before we get started and talk specifically about the lesson, I want to highlight our partnerships with OSU Research Faculty. We've been working for the last 32 years with OSU Research Faculty to develop content for K-12 classrooms that bring aspects of OSU research into those classrooms. And Dr. Stephen Giovannoni has been a partner for a number of years, and we've created a series of lessons that help to explain not only the research that he's doing in K-12 classrooms, but excite students about that research that's occurring. And this Bloom in a Bottle activity, I think, is a great way to highlight that. So now I'd like to introduce Dr. Chris Suffridge, and he'll be going over the lesson and the research connections with you today.
We're supposed to ultimately, okay, now we're recording. So you can come back and see this, but you should write down my email. If you have any questions about the science behind any of this, the experimental design, anything, please feel free to shoot me an email. I would love to hear from you. I always say this, no one ever emails me, makes me sad. But anyhow, I'm here to help. So moving forward, maybe. Are we moving forward? It's an interesting question. Yes, now we're moving forward. Wow, technology, sorry. Okay, so the expected learning outcomes out of this, I want you guys to take away the ocean is the largest biome on Earth, and it is full of life. Microorganisms are the most abundant and most important inhabitants of the ocean. Microbes are diverse in their size, shape, and function. Microbes form ecosystems where their interactions impact global processes, including the carbon cycle. And we can use models or experiments to study and these processes, and those processes can include the carbon cycle. So I like to start off my talks with the picture of the world, a map, and we have an arrow. We are right here in Oregon. And when you look at this projection of the way the world is, you're kind of posed with an interesting question, and a question that I think students tend to ask themselves: is the Earth blue or green? Is it primarily a continent-centered Earth, or is it primarily an ocean-centered Earth? And when you look at a classic classroom map, pull-down map type projection, it looks kind of like it's a continent-centered Earth. However, when we look at a realistic projection, now I have a Google Earth image. The arrow is still pointing at the Willamette Valley in Oregon. And it, again, looks relatively continent-centered. However, when we turn the globe just a little bit, we see all the continents disappear, and we really do, in fact, have a blue planet where 70% of the Earth's surface is ocean. The ocean is on average 4,000 meters deep, and the majority of the ocean is low nutrients, which means that microorganisms dominate in these environments. So every drop of the ocean contains life. And something that I really like pointing out is, so if you think about land, the other 30% of the globe, if you think about the habitable space vertically, you maybe have, I don't know, 10 meters above and a couple meters below the surface of the earth where things are living. You have trees and forests that go a little bit above, and you have some stuff in the soil underneath. So we can say, I don't know, let's say 50 meters over 30% of the globe. But on the ocean, you have 70% of the surface of the ocean, or 70% of the surface of the globe is ocean. And it's 4,000 meters deep on average. And every drop of that immense three-dimensional space is full of microbial life. So this is a huge scale issue. There's a ton of microbes in the ocean, which then feeds into this next question I guess I've answered for you. But what types of organisms live in the ocean, and what are most important to our planet? And I find that generally people that aren't marine microbiologists, when you say I work with the ocean, this is kind of where they go. You're a marine micro, or you're an oceanographer. You must study whales, sea otters, fuzzy things. And this isn't really what we study. These aren't the most abundant organisms on the planet. In fact, the organisms that we do study are in this picture. We just need to zoom in a lot further. And when we do that, we get an image like this. And what this is an electron microscopy image of seawater that has been collected on a 0.1 micron filter. And what you see here are all sorts of bacterial cells. And these bacterial cells have been retained on this filter. And these are the most abundant organisms in the ocean, and they are the most important organisms in the ocean. So, a fun fact, in 1 drop of seawater, or 1 milliliter of seawater, there are about 1 million bacteria cells. So let's go back to the 70% of the surface of the earth. So we have 70% of the surface of the earth, 4,000 meters deep. In every milliliter or drop of seawater, there are about 1 million bacteria, left to right, north to south, top to bottom. It's a lot of organisms. These are the most abundant organisms you've probably never heard of. So some quick vocab before we move forward. First off, oligotrophic. What this means, it's an area of low nutrients. So the vast majority of the ocean, especially the centers of the ocean, have very, very low nutrient concentrations. So coastal areas are considered eutrophic. They have high nutrients, but the middles of the oceans are very oligotrophic. And in areas of low nutrients, microbial life dominates. So moving on, we have phytoplankton. Phytoplankton are things like diatoms or cyanobacteria. And you can think of these as like the grass of the ocean. They create biomass from the sun. They photosynthesize just like land plants. And an example of those is Prochlorococcus. We'll talk about that in a minute. On the other side of this, we have bacterioplankton, which use the biomass that are produced by phytoplankton, and they use those to survive. An example of this is SAR11. And then we have zooplankton, which are the bigger microorganisms, and these consume both phytoplankton and bacterioplankton, also some single cellular things as well. But these are, an example would be copepods. If you guys watch SpongeBob, Plankton in SpongeBob, the proprietor of the chum bucket, he is a copepod. So there's actually, SpongeBob was created by a guy that worked at a science center. So the animals in it actually are kind of decent the way they interact. But plankton is a copepod. And they actually look like that. They have one eye. So moving on, SAR11 is the most abundant bacterial plankton in the ocean. It was discovered in our lab a couple decades ago by Steve and colleagues. And this organism, it's a heterotrophic bacterial plankton. You can see these are 2 different images of it. It's these little semicircular guys on this filter. This is a zoom in on that image I showed you earlier. This is a membrane filter. The holes in that filter are 0.1 micron. You can see SAR11 is kind of going through it. These are incredibly small cells. They are the most abundant type of bacteria in the ocean, which I think we can say makes them the most abundant organism on this planet. They're responsible for somewhere between 6 and 37% of oxidizing the carbon in the ocean. So it's a huge amount of carbon that they process. They have very, very small genomes, and they're tremendously successful. It's a really, really interesting cell type to study. And that's kind of what we do here in the Giovannoni lab. So this is the most abundant type of bacteria plankton, and they're heterotrophs. They eat carbon that's been fixed by phytoplankton. So on the other side of this, we have Prochlorococcus, which is kind of SAR11's dancing partner, if you will. SAR11 is a heterotroph. Prochlorococcus is a phototroph. It fixes carbon like grass or trees through photosynthesis. And it's a very important type of phytoplankton. It's about 5 times bigger than SAR11. We have SAR11 is about 0.1 micron, which is this bar right here. And Prochlorococcus is about 0.5 microns-ish. So if you lined up, to give that scale, if you lined up 200 of those in a row, it would be the width of one human hair. These are very, very small. However, both of these organisms, SAR11 and Prochlorococcus, are incredibly abundant in the ocean. You have somewhere between 100,000 and 1 million of them in every milliliter of seawater. Prochlorococcus, pretty much in every milliliter of seawater in the surface ocean. So the first, we'll say 200 meters where there's light in the ocean, everywhere. And SAR11 is abundant at roughly those levels pretty much everywhere in the ocean. It's an incredibly successful cell. So here's an example of some other microorganisms. We have this copepod down here. This is a multicellular organism. It's a little animal. These are some Rosiobacter. Up here we have Trichodesmium. There's a whole bunch of cells, bacterial cells that live together. Interesting thing about Trichodesmium, it fixes nitrogen. So organisms like beans that have root nodules that fix nitrogen in the soil. This is an organism that fixes nitrogen in the ocean. And then here are some types of diatoms. They're a little bit bigger, but they're kind of cool looking. Microbes are cool looking. But they're very diverse in size. And this is an important thing that I feel like gets glossed over, because to humans, anything that you can't see is small. So it's small, you don't really think about it. However, the scale of small is hugely different. So I want to orient you with this figure. This is one of my favorite figures. It comes from a paper by Zoe Finkel that came out in 2010. On the top axis is in micrometers, so microns. And on the bottom axis, it's in meters. So cool thing with the metric system is they scale the same way. Microns are 1 million times, so 1 micron, there are 1 million microns in 1 meter. So it's 1 million times smaller than a meter. But, so down here we have 0.1 micron, or 0.1 meter. And up here we have 10 to the 4th, or 10,000 microns, or about 10 kilometers up here. So we see that on the bottom axis, we go from something that's about a fish size, it's about, I don't know, less than a meter, orca whale, which is, I don't know, about 10 meters, factory, then something that's as tall as the Eiffel Tower, and something that's as large as Manhattan, multiple kilometers wide. So this is a large size variation. But if we go into the meter space, we see we've met Prochlorococcus, and SAR11 would be down here, even smaller than Prochlorococcus. And then we have up here Trichodesmium, which I showed you in the previous slide, that guy. The difference in size between Trichodesmium and Prochlorococcus is the same difference in size as the entire borough of Manhattan versus a fish. So there's massive differences in the sizes of these organisms, even though they're all very, very small, which I think is really interesting and kind of cool. So what are these marine microbes doing? Why should we care about them? Are there any questions? I haven't actually looked at the chat at all. Jay, are we good? Cool. Okay, if you have any questions, feel free to pop in. But so what are these marine microbes doing? Why should we care about them? Oftentimes, I don't know, when I'm talking to non-marine microbiologists, they assume when I say I'm studying marine microbes, I'm studying bacteria that make us sick, like when you have sewage spills in the ocean, that sort of thing. That is a microbial process when human waste is released into the ocean, but that's not what we're talking about. These microbes are natural, they're always there, and they're incredibly important to the globe. Mainly, 50% of all the oxygen we breathe is produced by microbes in the ocean. And that's just not 50% of the oxygen we breathe here in Oregon, a coastal state. That's 50% of the oxygen over the entire globe that's produced is produced by microbes in the ocean, by organisms like Prochlorococcus or diatoms. That's really cool. They're important. I like breathing. Let's see, hide this. There we go. So how does this work? Photosynthesis by phytoplankton removes carbon from the atmosphere and it produces oxygen. Same process that trees and grass use. And here's the equation for it right here. We take carbon dioxide from the atmosphere, we take sunlight, phytoplankton then convert that carbon dioxide into biomass, sugar, and they release oxygen as a product. And you can kind of see we have this food web schematic down here where we have phytoplankton, they use nutrients, bacterial plankton then feed off phytoplankton, zooplankton feed off them, and we have this system that's going on. But at the same time that photosynthesis is going on, there's a competing process, respiration. So heterotrophs, and actually phytoplankton too, are respiring at the same time that photosynthesis is taking place. And it just so happens that the respiration equation is the exact opposite of the photosynthesis equation. You take reduced carbon, sugar, you use oxygen, you burn this sugar, and you release carbon dioxide. So in that case, photosynthesis is producing oxygen and sugars, and respiration is using sugars and oxygen. So, question you might ask, if you're looking at this and you see these two equations and you line them up like this and you sum them together, they sum to zero. So how is oxygen produced if photosynthesis and respiration are balanced? Interesting question, right? So the way this works is through a process called the carbon pump, and this is why we get really excited about this in our lab. The carbon pump sequesters some of that fixed carbon, allowing net oxygen production and net carbon dioxide removal from the ocean. And the way this works is, yes, those equations are balanced. However, the rates in those equations don't have to be balanced. So as phytoplankton grow, they're fixing carbon dioxide and they're releasing oxygen. However, as I told you, the ocean is this three-dimensional space, right? And things sink. If you've ever been on a boat or down by the ocean and you dump something overboard, it sinks. So as the phytoplankton sink, they take that carbon they've fixed with them. And as they sink and take that carbon with them, that carbon is removed from the system. So you're left with a net balance of oxygen in the atmosphere, and that carbon is removed. So a really great figure from Warden et al. 2015, it was in Science, shows how marine microbial interactions control the strength of the carbon pump and kind of how this works. Again, so here let's look at this. We have the sunlit ocean on the top and the sun up here, and then we have the dark ocean on the bottom. And we have phytoplankton that take light and carbon dioxide and they make biomass, they make themselves. In the process, they release oxygen. As those phytoplankton grow, they produce what we call particulate organic matter, or POM. And this POM, and it can be turned into dissolved organic matter, DOM. This is used by bacteria and archaea and zooplankton and other heterotrophic protists as food. It forms the basis of the food web. So this carbon is generally, as if it's used in the food web, it's going to be turned back into carbon dioxide. It's going to be burned. However, some of this POM sinks. It aggregates and it sinks. And a small percentage of it sinks. And that's what accounts for the ocean's ability to absorb carbon dioxide. And that's why we hear about how the oceans are able to compensate and have been able to compensate relatively well for the past two centuries for anthropogenic carbon emissions. As humans have produced more and more carbon dioxide through burning fossil fuels, the oceans have been able to absorb this excess carbon. And through processes like the biological carbon pump, they've been able to pull that out of the system. And we haven't seen massive swings in the climate yet, because the ocean's been able to absorb excess carbon dioxide. So one of the big reasons why we study marine microbes, it's not necessarily because they produce 50% of the oxygen we breathe. That's a great hook to get you interested and to listen to these slides. But the real reason is the other side of that equation. We study marine microbes because we're curious about how they control the global carbon cycle and how they control the planet's ability, the ocean's ability, to take excess carbon dioxide in the atmosphere and sequester that in the ocean, and what's going on there, and how specifically, how the microbial interactions, the interactions between all these groups, affect the efficiency of this pump. So that's kind of what we do in Giovannoni Lab, and that's, I think, a pretty good introduction to marine microbes. Does anyone have any questions? That's the end of this portion of the talk.
Well, thank you very much, Chris, for that great introduction to the importance of ocean bacteria. And so I pose this question here, thinking about how right now are you teaching students about microbes in your class? And I have some examples of some things that I've seen done in the class before, but just trying to get some ideas of what you're doing as we go into a way that you can study aquatic microbes with your students at distance or in your classroom when we get back to face-to-face delivery. So please go to the chat and put that in. So if you haven't taught about microbes in your classroom, this is a good opportunity to connect into it through growing cultures. I'm assuming that's on some sort of media from surface bacteria. Math teacher never attempted this. I think there's some great math you can think about of exponential growth of bacteria. I mean, just the equation that Chris posed about trying to figure out approximately how many bacteria are in the ocean. What is that? 1 mil of seawater contains 1 million bacteria, and the ocean is 4,000 meters deep on average. And then if you had some sort of area, that would be a great way to connect to math. Linked into the carbon cycle, that is excellent. Biology will be next year. Let's hope we get back to normal for biology. Now, there is an opportunity here to work with your students in something that they can easily do at home, and we've put in a number of different ways to do that. My chat is like covering up my screen here. So we call it Bloom in a Bottle, and this activity was first piloted in the Giovannoni Lab a few years ago. We've done this with teachers from around the state and know that it works really well. We've set it up into two distinct parts. One is sort of setting up the experiment. So how can you get students going out, collecting water, and just trying to get a bloom or those bacteria and algae to grow within the bottles and the water the students collect. And the other part really focuses on the experimental design. So we're going to talk quickly about setting up your bottles. Now, if you go to our webpage for virtual teacher professional development, you'll see this document right here. This document's going to walk you and students through collecting water. This experiment relies heavily on a nutrient source that you provide the microbes in the water you collected so they grow quickly. And in the activity as written, it's used this liquid plant food, Miracle-Gro. It's really easy to add a couple mils of that to each bottle, and that really drives growth through those ocean microbes. And we know that not all students are going to have access to a liquid fertilizer or any other type of fertilizer to use in this process. And so Chris came up with a great way to basically provide nutrients without using liquid fertilizer. Chris, do you want to explain that?
Sure. So this is kind of a variation of what we would call a dilution experiment in oceanography. And we do this all the time out on ships when we want to study the natural growth rates of bacteria in the ocean. So when we do that, we don't want to add any extra nutrients because we want to see how fast the bacteria are growing by themselves. So what we do is we take a whole bunch of raw seawater we collect, and then we filter it using a filter like I showed you the pictures of, to remove all the cells from that water. And then we add back in a known amount of cells from the water, from the unfiltered water. And then we allow those cells to grow in the diluted the filtered water. So basically you take natural water, you remove the cells, then you add some cells back in and allow them to grow when there's that background level of nutrients. So you guys don't have filters, pumps, etc. So what we think could work and hopefully will work, a caveat, I have not tested this, is if you go out and collect a natural freshwater source, a pond, a river, creek, something, go collect that raw water. And we have the exact volumes here, but you take the majority of that and you boil it, you bring it up to a boil for about 5 minutes, and students could do this at home. You're going to kill all the cells that are in that water. And in the process, you're going to open those cells up and you're going to release the nutrients that are inside those cells back into the water. And you're going to basically make a nutrient-rich solution. So you then would allow that to cool. And then the water you collected that you didn't boil, in the thing here, it tells you exactly how to do that, but you would add back in a certain amount of the unboiled water to the boiled nutrient media, and then you would have growth that would occur. So that's it's all outlined here with replication and all that fun stuff.
And what I really like about this is I think most closely aligns with the actual research that's happening that the Giovannoni lab is actually doing when they're filtering water out in the ocean. So a really good connection to what scientists are actually doing, which is always, I think, engaging for students. So as we move on to part 2, it's really about experimental design and going through this process of developing and testing a hypothesis. And so this is broken down into a number of steps. I'm sure these steps you have covered. We're not going to spend a ton of time on each one of them, but here they are listed out for you. And then we're just going to briefly cover these because in order for students to be able to do this before the end of the school year, they really have to be able to get to this point where they're running the experiment. The gathering the data and doing some of the analysis, those can all be done afterwards because it does take these blooms to grow. It takes some time for them to happen. But these are the basic steps that they'll be going through when they do the experimental part of the lesson. So it all starts with observation here. A great quote by Sherlock Holmes: "I see no more than you, but I have trained myself to notice what I see." Students making observations. This is sort of the basis of all good experimentation. And in here we're starting with the idea that these blooms occur, and students will be able to see this if they go out to different natural water sources. They'll notice at different types of year, especially in stagnant water, they'll see different things growing and may actually witness some of these blooms. So why are these happening? And there's so many different questions that you can start to ask as you get to that point.
Hey, Jay, can I jump in for a second?
Yeah.
Can you go back to the picture slide?
Yeah.
Just brief what we're actually looking at here. So this is the south coast of England and the English Channel. And we're looking from space. I'm using a satellite. And this big discoloration we see is a bloom of microbes called coccolithophores. and they're a microbial eukaryote, and they have their shell, they have a shell that's made out of calcium carbonate, the same stuff the chalk is made out of, interestingly enough. So these blooms happen really, really frequently. And as they happen, as I told you, the cells sink and they're deposited on the seafloor. And then through tectonic action, those deposits are then uplifted. So right here, also on the south coast of England, you have these giant cliffs, the Cliffs of Dover, White Chalk Cliffs of Dover. These cliffs are fossil remains of these blooms. So the cliffs are made, chalk that you use is the fossil remains of organisms like coccolithophores, plankton, phytoplankton that have calcium carbonate shells. So if you were to go, and people do, into these chalk beds, you actually can see the microscopic fossils. They are, all it is microscopic fossils that have been uplifted. It's really cool. So kind of a macro approach of where it sinks and then eventually it gets re-uplifted. But that's all I have to say. Also, someone in the chat said they have a question about seawater and freshwater. If you're doing the boiling thing, it needs to be fresh water, because if you boil seawater, you're going to change the salinity, and then we're not sure quite what will happen. Something interesting will happen, but it won't necessarily be normal. If you're using Miracle-Gro, or as it's stated in the lesson plan, the actual chemical nutrient additions, you absolutely can use seawater. That's what we study. We're a marine lab. That's all I have. Go for it, Jay.
Excellent. So getting back to defining a question, and this I think can be some of the most most challenging part for a student, is to really think about and define a question that they can then develop into a hypothesis. And students can take a long time going through this process. It may be a good time to let them sort of think on this a while, because there's potentially some more time to do it. You're not in a traditional classroom, but using the guidelines laid out here of asking broad questions that are looking for the root cause of these initial observations. So again, thinking about those, the Cliffs of Dover, and thinking about the microbes there. I mean, that's an amazing observation, and then trying to link those two things together. These questions at this point should be open-ended. Questions should stimulate initial research online. Students, there's a huge amount of information online that students can explore with a wide variety of questions. We've put a few here, but I think there's many, many questions that students could start to ask. We have a larger list here. Now, as you read through these, you may say that, well, some students aren't going to be able to have the resources to study these particular questions, especially when you look at the way the lesson is written. There's some specific nutrient solutions that would be added to limit or increase particular nutrients in the experimental phase. But there's also many things that they can look at: light, salinity, temperature. All these are easy variables that they can start to look at. What are those effects on their bloom? So you can have them come up with their own question. You can seed questions to them to kind of help go through this process. And we provide a list here, as well as a list in the lesson plan to sort of get you started.
I think one of the really important parts about this experiment, or this setup, but really science in general, is the way science is taught in high schools, and then also in college for a large part. It's a lot of rote memorization, and it gets away from the fact that science as a practice is all about investigation. What do I do in my lab on a daily basis? We investigate questions. We ask questions, we have a natural curiosity, we ask those questions, we design experiments or field research to go out and identify answers to those questions, and then we move forward from that. So something that's really cool about this, I think, is that students, this is kind of a pick your own adventure deal. What interests you? Is the student interested in how fast do phytoplankton grow? Are they interested in how many nutrients they need? Are they interested in how much light they need? This really can be whatever they want to test. The key here is that they're only testing one variable at a time, because that's the way you understand what impact that one variable has, is by asking a question that's a very carefully thought out question so that only one variable is manipulated. So if you're asking what impact light has, you would give all of your cultures the same nutrients, but you would expose them on a windowsill or different parts of your house to different levels of light and see what impact light has. If you're interested in what impact nutrients have, you would put in different levels of nutrients, but expose them to the same light levels. Does that make sense generally? Cool. Question about the see from the chat with adding back distilled water. Yeah, that could work. You also could just not and see what happens. Something will grow.
So I have another question. I teach math, but I'm going to be teaching STEM next year, and so I'm really new to all of this. Very new to all of this. You said phytoplankton is so tiny. Will students be able to see this without a microscope?
Yes, absolutely. So that's the cool part of this, is we're creating a bloom. So microbes have this great tendency of once the conditions are absolutely perfect, they exponentially grow. And this is where actually you can bring math into this really, really easily, is looking at microbial growth. It's not a linear growth, it's exponential growth. So you can, it's the same exponential growth formula you'd use to compound interest in whatever bank account you wanted compound interest. But they grow exponentially. And as they grow exponentially, they'll get to a density that's high enough that the color of the water will actually change, and it'll change to be green. So when you go down to the coast, or you go to a lake and it's green, the reason it's green is because of all the phytoplankton that are growing in it. Whereas if you go to Crater Lake and it's this deep sapphire blue, almost violet, nothing's growing in it. It's very, very oligotrophic. So the color, that color spectrum largely has to do with the cells that are growing in it.
Chris, could I jump in?
Go for it.
Yeah, so it's this, Steve. It's also possible to turn your cell phone into a microscope, and depending on what cells you get, sometimes they are very spectacular and beautiful.
We have in the lesson, I believe,
Yes.
there are several apps for phones, not only to turn them into microscope, but also a colorimeter app that allows you to measure the saturation of the color. Because the other really important part that is, not only are we investigators and we ask questions, but we collect data, and then we use that data to come up with the answer. So the cool thing about this is using those apps to track the density of the color and how it changes day to day. You could actually plot that up. Again, cool math connection. Plot that up and extrapolate which one grew the most, which one grew the fastest, because the color is directly proportional to the amount of cells that are in there. And Jay, correct me if I'm wrong, we have those apps linked, right?
We have examples of those apps, and also examples of different things you can get as far as a small, they're wooden or paper apparatuses that really help to turn your cell phone into a microscope. And those microscopes do actually quite well of seeing these different phytoplankton.
Phytoplankton are actually pretty big compared to SAR 11 or whatever. So you can actually, with relatively limited magnification, see them pretty easily. And they're really pretty.
And then I'm also wondering, like, what the time frame is to see something. Is this like you started and then weeks later you notice something, and so you do something in between, or is it, you know, more immediate?
Largely dependent on the starting conditions and the incubation conditions. There's no right one answer to this. If it's a really rich culture with lot of nutrients and a fair amount of light, you could see a bloom in a few days. I mean, if anyone's had an aquarium, you know how fast the windows get all cruddy and you have to clean them off. If it's not, it could take as much as a month. So
when we did this with teachers before, we were looking at, and these were just natural freshwater collected and sitting in a window of a classroom. We were looking at about 3 weeks to get real noticeable growth. But it can definitely take longer, and it really depends on the types of conditions that you set it up. And I think we're coming into a good time of year where it's a little bit warmer. There's a little bit more light, and these are going to grow a little bit faster. So going forward here, just going to sort of run through these really quick. You want to have your students, after they come up with a question, have some sort of testable hypothesis. I think the important thing here is that this is a yes or no. This is a question with a yes or no answer. And so students are either going to confirm their hypothesis or not. And that is going to be the basis of what their experiment is going to look like. So you come up with a testable hypothesis. In this example, we're talking about the phytoplankton community found in seawater in Newport, Oregon is nitrogen limited. In order to confirm this hypothesis, the following conditions need to be met. The nitrogen treatment has to have more growth than the phosphorus treatment, and the nitrogen treatment has to have more growth than the negative control. Again, here, the idea of controls is another important learning opportunity for students of why do you have controls in an experiment. This, I think, gets to the really fun part around experimental design. And that experiment is going to be designed to test that hypothesis. And students may find that when they get to this point, they're unable to test their hypothesis. So they may have to modify that in order to meet their ability or resources to test it. And you want to really match that to the experimental conditions. So thinking about this, what are some important factors to include in an experiment? Quickly respond in the chat with some things that every experiment is going to need as you're working with your students. What would you want them to be thinking about?
And while you're doing that, just a comment. This is something we deal with in lab regularly. We'll have a really good idea, and then we'll realize that the way we want to investigate that either isn't practical or isn't a direct way to investigate that, and we'll have to stop and go back to the drawing board. Generally, this is the form of one of us has a really good idea, and we run into Steve's office, and we're super excited, and we say we want to go do this, and then he says, well, maybe we need to think about this a little bit more. It's a good idea, but let's think about it a little bit. So it's an important part. Students aren't going to come up with a perfect testable idea from the beginning. You gotta, this is a work in progress of how do we ask a good question that's going to get an answer that we actually are able to use.
So I heard in the chat a lot of really great things. Here is some things that we've come up with, and I think I heard all of them. Manipulate one variable. Chris mentioned that before, the importance of knowing if your experiment is having any effect. You only want to manipulate one variable, not multiple variables. Having a negative control is something that's going to give you no results, and the negative and positive control, where the positive control is going to give you a positive result. These are important to make sure that your experiment is working. And then finally, replicates. You want to be able to replicate these results over a number of different tests. And so we have put in here approximately 3 replicates per part of the experiment, and you'll see this in our experimental design. Any comments on other things that people should be thinking about as they're setting up an experiment, Chris?
I just want to clarify what the controls are, because that's always something that is I think sometimes difficult. A negative control is a no a no response control. So in this case, if you're using the boiled water idea, it would be 100% boiled water, because your assumption is that your boiled water is not going to grow. So you want to test that. You don't want to add any live cells back to it. You just want to make 3 replicates of just boiled water, put them on the shelves. And if it doesn't grow, then any response you see, you know, is the cells you added back in. But if the boiled water grows just as much as the cells you added back in those other treatments, you know that, well, you didn't actually kill the stuff in the boiled water treatment, and you aren't actually seeing an effect of adding the cells back in. If you're using Miracle-Gro or the chemical compounds that we put in the lesson plan, your negative control would just be seawater or regular water. So because you're going to see growth greater than that by adding in the different compounds. A positive control is a control where you know that it should work. If it's going to work, it's going to work in that control. And that the reason you do the positive control is you want to determine that there's nothing in your experiment that's inhibiting growth or killing your thing. You don't, maybe by boiling the water, you actually created a toxic substance that's killing everything. So for the boiled experiment, the positive control would be 100% not boiled water, and see what happens with that. For the fertilizer or the chemical addition, it's a high level of nutrients added into it, because you should see growth under those conditions. Are there questions on that? Good, cool.
As you're working through this with your students, I think this is a really good check-in point with them. You have a check-in point as they're coming up with their hypothesis to make sure that it's testable. You have a check-in point here around their experimental design. By checking this and making sure they have the right elements in their experiment is going to be really helpful, that students don't spend a lot of time setting something up that is going to give them challenging results to figure out. And so the experimental design table is a good way to go, something that students can share with you, and you can take a look at it to make sure that they're on the right track. And there's an example of this within the lesson plan itself. So just looking at time, I want to make sure we can get through the rest of these things. So we're going to move a little quick. Again, please put any questions you have down in the chat. So once you have your experimental design, that's been approved, you're ready to go. This is where the real fun begins of setting up and running your experiment and gathering some data. Again, we've outlined how to collect the water in the procedure that's on our website. But as you're doing this, you're going to collect the water from whatever fresh or saltwater source you have available to you. You're going to determine these experimental treatments. You've already had those because you've done your experimental design plan. You're going to make sure you label everything. The worst thing that could possibly happen is you set this up and then all of a sudden you realize that you didn't label anything, so you don't remember where things are. So labeling and organization is always something to be thinking about as students are setting these up. And then you add the different treatments, whatever those treatments may be. Here, it's around adding different amounts of nutrients, but it could be putting them in different locations based on light, putting them in different locations based on temperature. Whatever that variable is, you're going to add those treatments. And then you really want to loosely cap the container. Now, you saw at the beginning that they were using mason jars. It's definitely a great way to go, and it's easy to loosen the caps there. But you can also use just regular plastic water bottles or plastic soda bottles that have been cleaned out with the labels removed. But it's pretty easy to tighten those caps on really tight. And you want to make sure there's some airflow that's occurring there because that's definitely going to be needed for the bacteria to grow.
Yeah, remember they're fixing carbon dioxide. So if they don't have access to atmospheric carbon dioxide, they're not going to grow. Something else you could do, not necessary, but might help, could also be actually a really easy variable to test, is agitation. So some of these cultures you could just leave, set up, no, don't touch them. And then another treatment, you could every day tighten the cap, invert 2 times, loosen the cap, something like that, and see which ones grow better. The idea being, if you're agitating, you're going to be bringing in more gas, and you're going to be mixing them up a little bit. They probably would grow better agitated, but that could be something really easy students could test.
Great idea. So that now you're in this gathered data point, and there's a lot of different ways you can gather data. We recommend that students are making observations, writing their observations down, and these observations could be on color change, it could be on different variable changes or other things they notice. Most students have the ability to take pictures with a cell phone. So taking a picture is using some sort of white background, like a piece of paper, white paper behind, to provide a consistent background can really help. And here's where you can start to use some of those other apps that can take technology that we already have or that students have access to or may have access to really get a sense of what's happening. And the colorimeter apps are great because they'll compare the colors of the pictures and really see some color changes that especially as those blooms are just happening, that is hard to see by the naked eye. There's microscopy that can happen. And I see filtering in here. What is filtering is filtering out part of the water to see what's growing in there?
Yeah, so the idea there is that at the end of the experiment, when you've decided it's done, you just use a coffee filter and filter each experiment onto a separate coffee filter and look at what is left behind and kind of get an idea of, I mean, if you really wanted to get very specific, you could filter the biomass out on the coffee filter. The phytoplankton will be big enough, they'll be retained on that filter. You actually could dry it out and weigh it and get dry weights. You could do wet weights. You could do something like that. Which treatment produced the most phytoplankton as retained on a coffee filter?
So we only have a couple minutes left, so I'm going to roll through these really quick. Here, this part, if you're doing this over throughout the end of the school year and into the summer, students are going to be doing a lot of this on their own. So giving them some support of how they're going to be able to draw some of these conclusions and determine if they have confirmed their hypothesis or not, and how they would go and redefine that hypothesis and do another experiment. And I think this is the really important part down here, is the repeat. Science is an iterative process, and you're constantly gaining new information and then repeating these different experiments. So this is something that students could do over and over to really determine what is happening and further refine their hypothesis and answer their initial question. Just a couple final thoughts here. You know, this is sort of the end. If the data supports the hypothesis, great. You've used a scientific method to determine the likely cause of an observed event. If the data does not support the hypothesis, also great. I mean, this is an important part. There's no losing here. You have determined what is not going on in using the scientific method. Repeat the process to investigate further. So there's always different options here, whether you confirm your hypothesis or not, it doesn't matter. You're always learning things. And so finally, there was an experiment just to show you some examples, and you'll be able to see this example in the lesson plan, where they were looking between nitrogen sources between freshwater and seawater. These were grown in these larger mason jars. This is day 1. You'll notice here on day 8, they're already seeing growth. You can see down here at the bottoms the growth, and the pluses sort of are talking about the growth here. And then finally, this is an older experiment here, but you can see how green these will get as the bloom actually occurs. So being able to see them is fairly easy with the naked eye. So you're being able to see them actually at about 8 days, and then here is 3 months. So the growth is pretty dramatic and continues to increase over that time. So with that, we're out of time. If there's any final questions, feel free to ask them. I really want to thank Chris and the Giovannoni Lab for supporting not only our virtual professional development, but for continuing partnering with us and developing really great activities. Please check our website. We have sessions every Tuesday from 10 to 11, and we even have one coming up on June 4th from 12 to 1 talking about food safety. So check that one out as well. If you signed up through Idealogic, you're going to get a PDU. If you don't receive one, email it us, pre-college programs. Any final questions?
Someone asked where they can find this lesson.
Where you can find the lesson here, let me just real quick move you over here. Can you guys see this now? So here is our website for virtual teacher professional development. You can see all the ones that we've already done. Here's our Bloom in a bottle, has the lesson plan, the procedure. Pretty soon there's going to be this PowerPoint that you could refer back to, as well as the recording of this session. So all those resources will be up here. And again, check out some of the other ones we did. We did a systems thinking lesson last week. And you can see all the different resources around systems thinking here. Next week, we have food illness outbreak and prevention and detection. This is a really cool activity from Ag in the Classroom that uses an online simulation to track down a foodborne illness. So something that you're definitely not going to want to miss will be here next week. Any other questions?
I also dropped my email in the chat. Please feel free to email me. I can set up a Zoom with you or frankly, even your students in your classroom if you want to do that. But I'm here. So
definitely take advantage of Chris. He's been great to work with and great with students as well. They set up a whole microbiology lab for a bunch of students we brought to campus last year. So again, thank you very much. And with that, we'll see you next week.